Plate counts. The most frequently used method of measuring the bacteria population is the plate count method. An important advantage of this method is that it measures the number of viable cells. It has a disadvantage like it takes some time, usually 24 hours or more, for viable colonies to form. So this can be a serious problem in some applications such as quality control of milk, when it is not possible to hold a particular lot for this amount of time. So that is the disadvantage of this method. So plate counts mainly assumes that each live bacterium grows and divided to produce a single colony. This is not always true because bacteria frequently grows linked in chains or as clumps. Therefore, a colony often results not from a single bacterium but from a short segment of chains or from a bacteria clumps. So to reflect this reality, the plate counts are often reported as colony forming units. When a plate count is performed, it is important that only a limited number of colonies develops in the plate. When too many colonies are present, some cells are overcrowded and do not develop. So these conditions causes inaccuracy in the counting of the microbes. So the Food and the Drug Administration, that is FDA, convention is to count only the plates with 25 to 250 colonies is reported. But many microbiologists prefer plates with 30 to 300 colonies. So to ensure that some colony counts will be within this range, so the original inoculum is diluted several times in a process called a serial dilution process. So see in the figure. So this is how we will conduct the serial dilution process. Let's say, for example, that a milk sample has 10,000 bacteria per milliliter. So if 1 ml of these samples were plated out, there would be theoretically be 10,000 colonies formed in the Petri plates of the media. Why? Because means there is a 10,000 of bacteria cells are there, so they will give obviously 10,000 colonies in the Petri dish. So this would not be produce a countable plate. Why? Because means it is impossible to count the 10,000 at a time. So in order to decrease the count of the colonies for a perfect and accurate calculation, we will prefer this serial dilution. So here in serial dilution, if 1 ml of these samples were transferred to a tube containing 9 ml of sterile water, that is simply diluting with 9 ml of water, so each milliliter of liquid in this tube would now contains 1000 bacteria. If 1 ml of these samples were inoculated into a Petri plates, there would still be too many potential colonies to count on a plate. So we need to furthermore dilution. So therefore another serial dilution could be made. That is 1 milliliter containing 1000 bacteria would be transferred to a second tube of 9 ml of water. That is second dilution. So here each milliliter of this tube would now contains only 100 bacteria. And if 1 ml of the content of these tubes were plated out, potentially 100 colonies would be formed. So an easily countable number. So learning how to do serial dilution is an important part of certain experiments in microbiology laboratory classes. So the dilution will be made for 1:10, 1:100, 1:1000, 1:10,000, 1:1 lakh. So the dilution should be made up to 1:1 lakh in order to make easy count for the bacteria. So here there is a small calculation for counting of the bacteria. After forming the colonies, we will count the number of bacteria by using this formula. That is, number of bacteria per ml is equal to number of colonies on a plate into reciprocal of the dilution of the sample. This is how we will conduct the serial dilution. A plate count is done by either the pour plate method or the spread plate method. The pour plate method follows the procedure shown in the figure here. Here either the 1 ml or 0.1 ml of the dilutions of the bacteria suspension is introduced into a petri dish. Here the nutrient media in which the agar is kept liquid by holding it in a water bath at about 50 degrees centigrade is poured over the sample, which is then mixed into a media by gentle agitation of the plate. When the agar solidifies, the plate is incubated. With the pour plate technique, colonies will grow within the nutrient agar as well as on the surface of the agar plate. This technique has some drawbacks because some relatively heat sensitive microorganisms may be damaged by the melted agar and will therefore be unable to form the colonies. Also, when certain differential media are used, the distinctive appearance of the colony on the surface is essential for the diagnostic purposes. So here the colonies that form beneath the surface of the pour plate are not satisfactory for such tests. So to avoid these problems, the spread plate method is frequently used instead. So here the 0.1 ml inoculum is added to the surface of the pre-poured solidified agar media. The inoculum is then spread uniformly over the surface of this media with a specially shaped sterilized glass rods. So this method positions all the colonies on the surface and avoid the contact of the cell with the melted agar. So this method will give easily countable colonies. So this is about the method of preparing the plates for the plate counts. The first one is pour plate method and the second method is the spread plate method. Thank you for watching this video. Please subscribe to our channel for more videos.